Hi, everyone. I don't know if you can hear me properly. So it's 4 and I want to start on time so all of our speakers have time and we don't have to rush into questions. So I'm Gina Kardik and I'm from the European Forum of Farm Animal Breeders, and I would like to welcome you to a series of webinars on decoding genome in monogastrics. And this is from the project Gene Switch. And so just as background, the European Forum for Farm Animal Breeders is actually the voice of livestock in Europe. And we have different members from different sectors. And through those sectors, we're able to participate in different projects in Europe, which include Gene Switch, but also with other species, which is poultry, pigs, ruminants, insect and aquatics. And we also have a sub-entity, which is FabreTP, which is the technological platform. And this regroup breeding and reproduction. And in this platform, it's not only the breeding sector, but also universities, knowledge centers, and together we participate in those projects. And so before starting, I would like to mention some house rules. So everyone is automatically muted and you will be able to speak and ask questions. And my colleague Danilo will be able to unmute you and allow you to ask questions. Thank you, Danilo, for helping on this. And the next part is I would like to invite Natalie Schweiger, who is from EMABG, and she will introduce the program and she will discuss a bit about what they're doing. Thank you and welcome.
Yeah, it's I can already talk, right, Gina? Okay. Yeah. Hello, thank you for the introduction, Gina. My name is Natalie Schweiger. I'm located in Vienna. We are at the University of Natural Resources and Life Sciences, Vienna, and I'm the administrative coordinator of the study program called European Master in Animal Breeding and Genetics. In short, it's EMABG. And this is an Erasmus Mundus master program where students can get scholarships for their studies. It's a high, top joint master degree program, so that is tough application phase, I would say, selection of students. We are a consortium of 6 European partner universities and 3 academic associated partners. And also within the consortium, we have 13 associated industry partners where students can go for their master thesis or internship periods. The European Master in Animal Breeding and Genetics, you can go back one slide, sorry. It is, it responds to the current and future needs of the animal breeding sector and includes also the industry. And it's we are looking at real world applications and also focusing on including internship placements and topics at industry partners. And you can find all our information on emabg.eu. And yes, and here we are showing our consortium partners. So in Germany, we are University of Göttingen. Then in the Netherlands, Wageningen University, and in Norway, Norwegian University of Life Sciences, in Sweden, Swedish University of Agricultural Sciences, in France, AgroParisTech at Université Paris-Saclay, and Boko University, where the coordination of the master program is taking place. And then also below, you see a list of our industry associated partners, which I will not name, but if you want to know more, you can find them also on our website. And on the next slide, you see our study track structure, or program structure, which shows the complete year of studies of an EMABG program. So they are students, sorry. So they are starting with a welcome course where all of the students are meeting. This now takes place at Boku University, and then they start at either Wageningen University or University of Göttingen in Germany, and where they also do a breeding lab activity or an internship period during their first year. And then after the first year, they move to the summer class where they are meeting together again, and also meeting the students of the first year. And afterwards, they are moving to their second year university where they are then doing also their master thesis research and other courses. And they finish by a joint graduation ceremony at Boku University again. And if you want to know more about the program or the application or admission criteria, please feel free to visit our website, emabg.eu, or you can also send me an email. So I'm happy to answer your questions. Thank you.
Thank you, Natalie. So this was a quick welcome and introduction from both EFAB and EMABG. I would like to thank both our speakers of today, Hervé and Sébastien, who will provide us with the knowledge and expertise on the project GeneSwitch, but also on genome and annotation. So I will leave the floor to Hervé, who will have 15 minutes to present, and then we'll have 5-minute round of questions. So please feel free to ask questions. There's no wrong question. There's always good questions where we learn all together. And I'll leave the floor to Hervé now. Thank you.
Okay, so thank you, Gina. So I will present the GeneSwitch project. I'm Hervé Acloque. I'm working in France at Juan-Josas on animal genomics, and I also try to develop new in vitro system for phenotyping. So the GeneSwitch project is funded by the European Union Commission and is coordinated by Elisabetta Giuffra, and I'm the deputy coordinator. And it's included in the FANG initiative. So FANG is named the Functional Annotation of Animal Genomes Initiative. It's a global group of people that want to improve and to accelerate the work on genome to phenome. And for that, we want to provide to all the scientific community functional maps of most of the animal genomes that are not model animals, so livestock species, but also companion animals. And we try to do it by looking at different times, different type of cells, different tissues, different environmental challenges. And after that, the interest of getting this is to transfer this information either for fundamental research, but also for applied science, for biomedical purposes, but also for animal production, for example. So just to show you a bit the contributors of the project, so most of us are either in Europe or in North America, but we have also collaborators from China, Australia, Africa, Middle East, and South America. And the idea of the project is really to get open sciences. So all the things we do are reusable, provided with rich metadata, so you can work again with the data sets we produce and you know what we have made on it. And also to try to get some standardization of on the methods we are using. All the things we are producing are on this data portal named data.fang.org, and you can easily get it. You can go, and it's, I think, not so complicated to navigate inside and to get information. So GeneSwitch is within the FANG initiative, but it's a European project with 11 partners, most of them from the west of Europe. So we have people from UK, Spain, France, Netherlands, Belgium, Germany, Italy, Sweden, and some companies also that are contributed to the projects, and most of them are breeders. So the idea of the GeneSwitch project is to look at functional annotation during developments because actually it's difficult to link the information we get from the genome and the information we get from the epigenome to understand really how the genome is working, how genes are expressed, and how the phenotypes are built. And for that, we need to transfer this information into functional annotation. And this functional annotation will help us to understand better key features that are conserved between the evolution, during the evolutions, and that are key to build organisms, because we all know that either being a human, a pig, a chicken, we have more or less the same organs. We are a similar timeline of developments. The time can be different, but the main structures are conserved. So this is an important way to look at by looking at functional annotation. And the other point is to understand the differences between the species, but also the differences between the individuals, and also the differences that are linked with changes in the environment. And that's how we name, how build the phenotypes we are. So I will just share a movie. I need to keep the sharp. Oh.
Animals around farmed animals by unraveling the connection between the genetic makeup of animals and their traits. Across different climates and agrosystems, animals display different physical characteristics, allowing them to be adapted to different environmental challenges. Variability in DNA is one of the things that causes this variation, together with the environment. The complete set of DNA of an organism, the genome, is made up of approximately 5% genes and 95% intergenic, also called non-coding regions. These intergenic regions, including regulatory elements, modify how and when a gene is switched on, which can strongly influence traits that are important for welfare and production. We know that the regulatory elements play an important role, but we are not sure in what way, and how they are in turn influenced by epigenetic changes coming from, for example, the environment or food intake. This is why developing an encyclopedia for this 95% of the genome is so important. This encyclopedia is called Functional Annotation. Functional annotation allows scientists to map the coding and non-coding features of the genome. With this information, animal breeders can select and breed animals that can cope better with a changing environment and meet societal expectations. The Eurofang initiative is part of the Worldwide Fang Consortium and was initiated by 3 Horizon 2020 funded projects. Eurofang continues to grow and now comprises more EU-funded projects. For more information, visit the project websites and follow the Eurofang social media.
Okay, so it was just a summary of what I said before, but it's a movie by a joint initiative that clustering all the European projects on animal genome annotation. So now coming back to gene switch, what we wanted to do is really to compare in pig and chicken how the early morphogenesis and late morphogenesis is affecting gene expression by looking at switches in gene expression, but also in the activity of regulatory elements like promoters, enhancers, and how this can be affected during development. So the first aim of the projects and what we are going to speak today in this webinar is how to identify the functional elements in the genome of these 2 species, how to identify the dynamics of these elements during tissue differentiation, and how they can be conserved or different across species. And finally, the aim of the project is to give to the scientific community a high quality, richly annotated genome annotation maps. The second aim of the project that will be discussed later in 2 other webinars is to use this information either to improve the software and the algorithm we are using to do genomic selection and to improve the prediction accuracy of breeding values in commercial populations. The second one is to look how we can modulate the epigenetic informations by the diets in order to get new tools for farm management, because working on the diet is an easy way to transfer it to farms. Finally, the third aim of the project is really to provide a standardization of data and processes that allowing people to reuse, to find, and to access the data we are producing. So I will detail a bit more now in the 5 next minutes how we do that to GeneSwitch. So in GeneSwitch, we have recorded all the metadata we are producing regarding samples, experiments, and analyzing. And all these metadata are available on the Func data portal, the one I mentioned before. It includes protocols, so you can find all the protocols we use for all the molecular assays we designed, but you can also find information on the samples we are using and all the experimental and analysis parameters we use, either for the primary analysis or the secondary analysis of our data sets. Of course, all the raw sequences have been deposited in the European Nucleotide Archive, and the pipelines are also publicly available either on GitHub or NFCore. So working on this, we can say that all the information and material we are producing with GeneSwitch can be easily findable, accessible, interoperable, and reusable by the scientific community. Just some examples. So this is the metadata you can have for one chicken tissue. So you can have the information of the birth date, where it was bred, the weight of the animals, the type of breed or lines, the embryonic age, the project, and the sex of this animal, and so on. And after you can come back to the sequences that have been produced with this sample, and you know that they are extracted from a chicken embryo of 8 days, and it was a male. The same for the pipelines. All the pipelines are easily accessible, either on NFCore or on GitHub, for all the molecular assays we have done. All the information is also accessible on Ensemble, on a dedicated web page for the GeneSwitch project. You have the directions there. And you can look at the tracks corresponding to the gene expression data sets. And so you can see here, for example, the different isoforms we can observe with our data sets and if they are present or not in the different tissues. What we are going also to do with EMBL-EBI is to produce a new regulatory build. So the regulatory build is something that integrate all the regulatory information, so you can find promoters and answers if they are active in one tissues at one stage or in different tissues at different stages. And this is really a powerful foundational resource to enable and accelerate research, either for academic labs, but also for the industry. In addition, we have also a specific web page in the Func data portals, where you have exclusively the data sets produced by GeneSwitch. So you can find all the studies we made, either for the IC, small RNA, ATAC-seq, methylation studies, ISO-seq, and so on. And so you can download these data sets and you can reuse these data sets following the Func rules of sharing data. We are currently developing a new tools that allow to see, for example, chip-seq information. So this is something you can load on one experiment. So this is the H3K27 acetylated chip-seq in the cerebellum, and you can see the enrichment around the TSS in this case. So you have a brief view of the quality of the data, and after you can come back and download these data sets if you want to work on it. So this is the end of this first presentation. I think I'm on time, more or less. So I want first to thank all the contributors to the project. This is a huge project, and I'm happy to answer your question if you have some at this step. I will do another presentation just after on functional annotation. And thank you for your attention.
Thank you, Hervé. Does anyone have any questions regarding GeneSwitch and Eurofang or the FANG project? If you don't feel comfortable talking, you can also write your questions in the chat and we can answer them there. So if there is no questions, I think Hervé will continue with the first step of the web series, which is what is annotation?
We have a
There's
question.
one. Yeah, there's one question.
Sorry.
It came late.
Hi, good afternoon. I was just wondering, like, if it's also possible for like master's students to reach out to the ongoing projects and also possibly ask for maybe a good topic for thesis, which is also like part of the project.
So this is a good question. So actually the GeneSwitch project will end this year, but we have a lot of data now and data sets that we are still working on because it takes time to produce all this information and it takes more time to analyze and after to interpret this information. So I think it's a good moment to apply for PhD and to work on this all the information we have accumulated during these years. You can, if you want to do a PhD, you can write to the different partners and to ask whether it's possible to do. So it will be more dedicated to data analysis rather than experimental work. But yes, I think we are open to PhD proposition, of course.
Yeah, thank you.
So, and this will be the next presentation. So
yeah,
I will try to share again. So you can see it.
Perfect, Hervé.
Okay, so initially this presentation would have to be done by Jenny DeVos. So she's a PhD student in Wageningen. And she made her PhD on DNA methylation and the integration of the GeneSwitch datasets. So she's not available today, so I will replace Jenny, but I will mostly present her work. So she's going to finish now. She has done all her PhD on the GeneSwitch project. So it's a good example of what you can do working on this type of project and data. So I have also a short video showing where she's going to explain a bit why she decided to do a PhD in this field and how she's doing it. I'm sorry she's not there, but anyway, the video will substitute a bit the absence of Jenny.
My name is Ron Post, and I'm a PhD at Wageningen University in the Netherlands. My PhD forms a part of the GeneSwitch project. This is a collaboration between different labs, institutions, and the industry. We want to characterize the genes in these two monogastric species. If we look at the dynamics, we have to think of them as switches, like lights, where you can turn the light dimmer or brighter. And in this way, these genes are more expressed or less expressed. And we want to look at these in the embryo, in the chicken, and the fetus stage in the pig, and compare this to the adult species. Using a bioinformatics approach, I am developing a pipeline for the analysis of methylation data. I will explain in more detail what this data is in the second video, together with what the functional genome is. This pipeline will be used to analyze raw data and then receive useful results such as figures, statistics, and graphs. This and other pipelines will be used for the analysis of data from the project to predict regulatory elements, as well as tissue and species-specific patterns of gene regulation and comparisons between tissues and species. This is similar as to what I explained with the light switch and these genes. I will also explain more about the regulatory elements in a second video. This PhD will combine the fields of bioinformatics and epigenomics to contribute to the knowledge of this subject.
Yeah. I'm coming back. Okay, so this just to show you a bit what can be a PhD as you are master students. So you can see a bit what is our job day by day. Now I will move on functional annotations. And the first things I think we have to know is a bit what is epigenetics. Because to identify regulatory elements in the genomes and so to do functional annotations of the genomes, so we really need to understand what is information that is over the DNA. And so you all know that genes are expressed, but sometimes they are on, sometimes they are off. And it's not only a way of the operon, like in bacteria, but it's more complex in eukaryotes, because we can add different types of molecules, and it can be methyl groups that are added on the DNA, so what we name DNA methylations, and they are linked with gene expression, but it can also link with what is over the DNA, that is, which are the histones. So you can add also different chemical marks on the histone, like acetyl groups, methyl groups. And these histone marks are modulating the affinity of the histones to the DNA. And so finally, if you have a lot of acetylations on the histones, you are going to change the charge of the histones, and they will go back. They will leave the DNA. And so the DNA is accessible. And the accessibility of the DNA is really important because it's something we have to measure, because when the DNA is nude, it seems that it is highly regulatory. So that's what we are looking at by using ATAC-seq, for example. But finally, it's important to see that it's an important definition and epigenetic information we can get whether the DNA is nude or not. And why? Because when it is nude, other proteins can bind to it, either to activate transcription, like the RNA polymerase there, but also other proteins that will enhance the expression and the activity of the RNA polymerase. So finally, when the DNA is accessible, you can have the different type of molecules that are bind to the DNA and will facilitate the transcription of genes. And by doing this, you will accumulate RNA, more or less, and this accumulation of RNA will be translated to phenotypes after either in cell, tissues, or individuals. And it's known since years, but I think it's important to remind you what it is. And this is a clear example of the importance of epigenetic regulation for the building of phenotypes. So here it's the color of the coat of mice. When a region is specifically methylated, the color is gold. When the region is unmethylated, so the color is brown. When the region is unmethylated, the color is gold. So finally, the genome is the same in all these mice because they are isogenic. But by modulating the epigenetic information, you're modulating the phenotype. So it's really, it's not new, but it's really a key information you need to have in mind, mostly when you are working on functional annotation. And so just to summarize, so epigenetics is either heritable, either between cells or also between individuals that occur in the genomes, and they are not associated with DNA sequence alteration. So it's really epi information we are added on the DNA information. It can strongly influence gene expressions, and it helps to define what are regulatory elements and how these regulatory elements are being functional or not functional depending on tissue, time, stages, and so on, and the sex also. And finally, so yes, you will have this type of information that can be made between promoters, enhancers, and the proteins that are able to bind together. modulate gene expression. So now coming back to the functional regulatory genome and the functional annotation, it's really to understand the link between the regulatory elements we can identify, then the expression of genes that are linked to these regulatory elements, and after the traits of interest. So in our cases, in breeding, we are most interested in the production, like for eggs, meat, but also for the health of the animals, the well-being of the animals, the ability of the animal to be adapted to changes, like climate changes, environmental changes, and so on. And so it's really key to build finally the way we are going to select the animals for the future. So functional annotation is really a multidimensional approaches, because it depends on, I will just describe some, but we can always add more. So it depends on the type of tissues. The regulatory elements you can identify can be different regarding of the different tissues you are working on. They can be different in the brain, in the muscle, in the liver, the lungs, the small intestine, and so on. But the second point is that these regulatory elements in one tissues can be different during the lifetime, because you have first the building of the organs and what we name ontology. So starting from early embryogenesis to late embryogenesis, the new chicks, and after you have the sexual maturity and the aging. And all this lifetime is affecting the regulatory elements we are looking at. So you need to consider also this dimension. The third one is the phylogeny. We have, as I said before, less or more the same organs. Either we are chicken, pigs, cows, human, and so on. But you have also differences between these organs. And so you have conserved regulatory elements between species, and some that are in some way different. And that's also something we need to consider. And within the GeneSuite project, we are comparing regulatory elements that are conserved between pig and chicken, but also those that are different. And finally, and this is, I think, the idea of the third webinar, the environment can also affect the regulatory elements that are present in the genomes. And you can affect them either by modulating the nutrition, the climate, the circadian rhythm, the health situation, so the health challenges, for example. And all this can also activate or switch off specific regulatory elements on cell types. The last ones could be also the importance of the sex. So male and female do not have always the same active regulatory elements in the same tissues. And of course, the genetic variations, that will be more the topic of the second webinar. So how are we integrating all these dimensions to explain gene expression and the regulation of the genome activity? So I will just start with a small example. So this is a gene. This gene can be transcribed with different isoforms, so I use 1 or 2. And we can identify potential enhancers in the genome, a promoter, and other additional enhancers. And so when we look at the early embryogenesis, we can observe by molecular assays that the enhancer 2 is active, the promoter 2 is active, and the gene that is transcribed is the isoform number 2, for example, number 1. But if we only look at this stage, we will not identify the other regulatory factors. So we need to go deeper and along the time. And if you look at another stages or 2 other stages, you can see that we will detect these new enhancers and different isoforms corresponding to this gene. And if we look at the adults, again, we will perhaps identify another isoform, or we will quantify differently the different isoform and the activity of the enhancers. So finally, you can see in this short example that it's really important to integrate the different dimensions I said before, to really get an exhaustive view of the regulatory genome. So how we do that? So first, we need to identify the regulatory elements at different times and tissues in different species. And we also have to look at gene expression to identify isoforms, but also in which way the gene is active or inactive. And so for that, we have a lot of molecular assays. So I don't have time to really explain each molecular assays, but I can show you what they provide. So we can look at the conformation of the genome. So this is the spatial conformation, the proximity of DNA regions together in the nucleus by doing IC or ChIP-seq with the CTCF protein. We can look at the DNA accessibility with the ATAC-seq. So what I said before, it's really important to see when the DNA is nude and accessible for transcription or regulatory proteins. And that's why we are always doing ATAC-seq in our annotation experiments. After, we need to identify potential promoters, enhancers, repressors, and for that, we do ChIP-seq with specific histone marks that are known to be enriched either on promoters, enhancers, repressors, insulators, and so on. We also look at DNA methylation with these two assays, so whole genome bisulfite sequencing and reduced representation in sulfite sequencing that give an information on the levels of methylation of genes, promoters, and enhancers. And of course, we look at gene expression. And for that, we combine different types of gene expression methods. So we are looking at mRNA-seq to see coding transcripts, for example. Small RNA-seq to look at small transcripts that are less than 200 base pair. And ISO-seq to have a large view of isoforms that are expressed in one cell type or one tissue. And why we are doing that? Because as you can see here, we can see a dynamic that is common to DNA accessibility and gene expression. So this is a paper that I've looked at, I think, skeletal muscles in the pig. And you can see that the information we get from the ATAC-seq is different and follows the development of the pigs, starting from the fetus of 45 days, 60 days, 90 days, 1 week, and 6 weeks. And it's exactly the same for the gene expression. So if you look at the profiles of gene expression, you can see that you see the same dynamic starting from D45, D65, fetus of D65, day 90, 1 week, 6 weeks, and so on. So it's not causality, but it's a correlation that the regulation of the epigenome and gene expression is strongly linked. And that's why in the GeneSuite project, we decided to compare the epigenome and the gene expression at 3 different stages in 2 species, the chicken and the pig. So we looked at early organogenesis, late organogenesis, and newborns, and in different tissues, like those from the endoderm, the liver, the lung, and the small intestine, those from the mesoderm, so the kidney and the skeletal muscle, and those from the ectoderm, so the cerebellum here and the skin. And we have done all these molecular assays I said before. And after producing the data sets, we have done the primary analysis, working on pipelines we developed or we use, and which are available online. And so the first result that is really strange and interesting. So when you just look at DNA methylation in pig, you can see that for some tissues, the samples will cluster by tissues. So for all the liver samples cluster together, either they are from newborns or early fetuses. And it's the same for the brain, but it's totally different, for example, for other tissues like the lung, the small intestine, the kidney, or the skin, where samples are clustering first because they are from early embryos. And what is interesting is that we can see exactly the same in chicken by looking at small RNA-seq. So we are looking at totally different molecular assays, totally different information. And what we see is that for the kidney, the liver, and the cerebellum, the samples are clustering by tissues rather than stages. So you can see here that for the cerebellum, the 3 stages are clustering together, the same for the liver, and the same for the kidney. But when you look at all the other tissues, they are clustering first by stages, because perhaps the similarity at these stages for these cell types is more important than the tissues they are going to produce after. This is really amazing. The second point I want to comment is the work of Gianni on promoter and enhancer methylation. So usually we said that when a promoter is not methylated, the gene is expressed or can be expressed. When the promoter is methylated, the gene cannot be expressed. And it's the same for enhancer. So if you have a promoter with a strong methylation that is not methylated, and an enhancer that is not or low methylated, you can have the expression of the genes. If one is methylated or the others, you have a low or no expression of the gene. So Gianni, what she has done, she looked for unmethylated regions and low methylating regions in the DNA of the lung here, of the pig. And what you can see is that unmethylated regions are close to the TSS, and this is normal because we are looking at promoters, and the low methylated regions are more far away from the TSS, and it's also logical because they are targeting more a usual topic of an answer. After, what is of interest is to look at the changes and the dynamic of the methylation of regulatory regions. And this is really amazing because for the kidney, at the beginning of development, we have the mesonephros. And the mesonephros is not a functional kidney. It's a structure that is going to disappear to give rise after to the mature kidney. And what you can see is that you have a lot of regions that are fully methylated at early stages that are going to be low methylated or unmethylated. And the reverse is true. So you have, and this is really amazing for the kidney. And this is not the case, for example, for the liver. When you look at the liver, you have not so much changes, regions that are switches from unmethylated to methylated, or fully methylated to low methylated or unmethylated. And what is amazing is to see that depending on the tissue you are looking at, you will see strong differences between stages or not so strong differences between stages. And this is really true because the kidney is really a part during development from the other organs. The second thing Gianni has done is to look at genes that are highly expressed or not really highly expressed, and how the methylation is affected in these genes. So the genes that are highly expressed are here in pink or lila, I don't know exactly, pink. And you can see that they are really unmethylated in the promoter, but the strange thing is that are also strongly unmethylated after the gene body here. And that is totally different from the other type of genes, which are mostly unmethylated at the promoter level when they are mildly expressed, but they are strongly methylated at the end of the gene. So when the gene is not transcribed anymore. And this is really a specific feature for some genes. And then she looked at the genes that are within these features. And for example, for the skin, she saw that you can find genes that are really important for the physiology of the skin, like the retinol, the keratin, and the epidermal growth, and the metabolism of these cells. And so it gives some biological sense of this. And when she did this for the brain, again, you can see that you are affecting genes that are specific of dendrites, dopamine neurotransmitters, cell division, and also hormones. So this is really, so it's really amazing to see that and how the study of regulatory elements and the annotation on these regulatory elements links to genes that are really important for the function of these tissues. So after that, the idea is to provide an integrative analysis and to understand how with all this information we can explain gene expression and later phenotypes. So for that, we have to combine, for example, the accessibility of the chromatin, the information we have on the histones and the marks that are specific of these histones, either for promoters, enhancers, transcriptional activity, or genes that are silenced. And DNA methylation, as I said before, that's also regulating genes. And so when you combine this, what you look at are specific signatures that will give you information on the regulatory elements, whether they are coding for active transcription starting sites, open chromatins, something that is ready to be transcribed, but it is not transcribed, that remain bivalent or poised, and regions that are highly repressive or weakly repressive. And so it's some things we are doing now within the project. And soon we will be able to release some maps. And when you will look at the genes you have interest, or the regulatory regions that control the expression of these genes, you will get this information without to do again all these type of studies that are really expensive and difficult to produce and takes time to produce too. So finally, we do that because we want to provide reference for the future, so for new research that is not available or are different from model organisms. It will also provide us some dynamic changes that occurs either in the epigenome over times and development. Oops, sorry. And finally, to get insights into development and gene regulations and the importance of genes to regulate this switch between developmental stages, either being conserved between species or being specific of each species. So finally, and I'm a bit late, I want to thank all the people from the Gene Switch project, because it's a huge work to produce all this data, to analyze all this data, and to make them accessible to everyone. It's not easy. It can be easy to produce data, but to do it well and to get it accessible to everyone is not so easy. And for Gianni, I want to thank people from Wageningen University, Martins, Ole, and Martin, which have done all the work on DNA methylation and some of the work on data integration. So thank you all, and I will be happy to answer questions.
Thank you, Hervé. So there is 2 questions in the chat, but maybe some other people also meanwhile want to raise their hand and we can let them speak. So the first question is from Gladys, where she says, great talks, and if there will be future courses or training workshop related to the topic. So this, I think,
Yeah, Gladys, I understand your questions. The first idea was to go to the summer courses in Vienna, but we have, I think most of the people in the Gene Suite Consortium are family constrained, and it was difficult to do really training workshops. But I saw that Tatiana from ongoing European projects is there, and they will organize additional trainings and workshops to be able to work with these data sets and to learn how to do it. We have done some before, and perhaps with EMABG we can follow and to organize one day of trainings, either to how to use Func data using the Func data portal, how to use the pipelines we developed, and so on. Yeah.
Exactly. Thank you, Hervé. And the second question, I think you answered it already. Afterwards, it's from Samrawit, and she is asking, how did you measure the gene expression in addition to the phenotypic observation? And how did you know if the phenotypic variability is not due to TE rather than epigenetics? And I think she mentions that the first one has already been answered.
What do you mean by TE?
Samrawit. Transposable.
Okay, yeah. Well, the activity of T are mostly active, so they should be silenced by epigenetic marks in the genome. So by looking at epigenetic marks, we can see whether they are effectively silenced. And we did not do some germ cells functional annotation within the project. I think it's a lack. I don't know why we didn't do it, but it should have been of real interest. I think Sébastien can answer this type of questions because he worked on it previously. Yeah, phenotypic variability can be due to TE, but it was not the things we look at, so I cannot answer.
Yeah, just for the transposable element after. Yeah, we didn't made a real notation of them. It's it's a big subject, transposable element. But what I can say, it's just yes, some of them are expressing, because I found some repeated RNA sequences, gene annotation. So some of them are active, but just the subject of repeated elements is too wide for being treated right now.
And so for the last question, I let you read it. So was your research on a single generation, or did you look at transgenerational inheritance? So for the annotation, we don't do that because it's not the mean of the project. The mean of the project is really to get an exhaustive view of what can be active or inactive regulatory elements and to map it. But in the third webinar, you will have a study where they looked at the effect of diets in the mother and the effect of this diet on the piglets in the muscle and the liver. So yes, we look at transgenerational inheritance, but only on between the mother and the progeny. And it will be the topic of the third webinar, I think.
Thank you very much, Hervé. I think, as you must have, realized, there is a few other webinars coming. So I think Hervé mentioned 2 already. So now I will leave the floor to Sébastien from the University of Edinburgh, who will explain to us how to annotate a genome. Sébastien, I leave you the floor.
Yeah, just time to share. Excuse me. Yep. Here we go. So I'm Sébastien Guizard, so I'm a postdoc at the Rodin Institute. I have been hired on the GeneStitch project for annotating the genome of the pig and the chicken using one specific technology, which is called ISO-seq. But today I'm going to try to present you how to annotate a genome. Hervé talked you more about the post-analysis of the data. What I'm going to present you is to show you how do you generate this data, at least a very small part of them. As Hervé presented to you, in a genome we find a lot of things. So there is the genes, indeed, but as he explained, it represents a very small region of the genome. For example, the human genome is only 2% of them. But there is also, aside of these genes, there is also the regulatory elements, such as the promoters, as Hervé present, the enhancers that increase the gene expression, silencers. There is also repeated element, so like you can see here. And so the repeated elements can be mobile, so they can create a copy of themselves and integrate it somewhere else in the genome. And we can find also some structural variant, such mutation at the single position of the genome, which is called single nucleotide polymorphism, or SNPs. Or it can be a sequence that have been inserted between when you compare 2 individuals of the same species. So it's called insertion. And if the sequence have been deleted, it's a deletion. So you have all these differences that can be also annotated in a genome. And also in the biggest part of the genome, which is the non-coding part of the genome, and you can find a lot of things inside of it. There is a tRNA, miRNA, non-coding RNA, long non-coding RNA, etc., etc. And it's a huge part of the annotation. But today, I cannot present you all the way to annotate all of these things. So I'm just gonna try to show you how do you annotate genes and what are the methods that exist for doing that. So there is, for annotating genes, there is 3 different methods, which are called ab initio, homology, and pipeline. And I'm gonna explain you the advantages of each of them works and what are the advantages of each of them. So the first one, the first kind of method, it's the ab initio method, and it's the simplest one because for using this you just need one decent genome sequence. And I say decent genome because few years ago, most of the genome were just Swiss cheese with a lot of holes inside, very fragmented. Now it's more easier to assemble, so all the genome should be okay for running this. So how does it work? So those methods use mathematical probabilistic models. So there is a lot of them. For the most known of them, there is the hidden Markov model, the general hidden Markov model, weight array method, and so on. And the things for using these models, they need first to be trained, and they are trained using genes evidence, using evidence. And so what are those evidence? These evidence are in fact genic features. They can be related to the gene content. For example, you can use the common usage, the hexamer usage, GC content, compositional bias between codons, the position, the nucleotide periodicity, exon entrance size. So you can have all these characteristics of genes, but you can also use the signal around genes. For example, the promoters, the off, the start codon, the splice site, the stop codon, polyethyl, CpG ion, and so on. And using all this information, you are able to train those models, and they are capable of detecting those genes. So those methods have the big advantages to be very simple to use, because you just as I said, you just need your sequence, your genomic sequence. You run the program on it, and you have your annotation. The other big advantages of this, it's that they will create an exhaustive gene annotation. That means they will try to detect the complete population of genes in the genome. And if you are lucky, you will not need to train the model. But if you are unlucky, you will have to train the model, because those models are mostly trained for the reference genomes, the most studied genome. So if you are working on something very specific, you will have to produce extra work for training this model. Another drawbacks of this method is they tend to produce false annotation, false positive. So you should be very careful with your result and apply some filters to clean them. And the biggest problem of these methods is that they didn't detect the isoform. So I will talk about them, but just as a reminder, what is isoform? For example, if you have these genes, these genes with these 4 exons, it will be transcribed into mRNA, and this mRNA will contain these exons. But the set of exons using for creating this mRNA can be different. For example, here you have the 4 exons, but for these 2 different versions, you have the second exons that have been skipped, and here's the third exons that have been skipped. So here, with one gene, you have 3 different isoforms. So it's a big problem of this technology. So that's why the homology methods are better for that, because instead of detecting directly the genes in the genomic sequence, we will use the RNA directly, sequence the RNA for detecting the genes. So the first step is to sequence the RNA, and then we map them on the genome for locating them. So about the sequencing of RNA, a long time ago, when I say a long time ago, it was 15 years ago, sequencing was not so easy, not so cheap. So we didn't have so much of RNA data. So it was mostly express sequence tag, EST, and cDNA, which are, for make it simple, very fragmented RNA sequence. But now that sequencing is cheap, very cheap, we can, sorry. Excuse me for this intervention. What I was saying, yes, the sequencing is very cheap, so we can produce a very high number of sequences. So we can afford to sequence the complete population of RNA in the genome. And so for doing that, for doing this sequencing, today we have 2 main sequencing technologies. The first one is the short reads, which is handled by the Illumina company mostly. And with this technology, so we will generate a very high number of very small sequence of around 250 bases. And the advantages of this technology is it produces reads with very few error of sequencing inside of it, and in a very high number of sequence. And the last advantage, it's very cheap, so we can afford to do several of them. But in the opposite of the short reads, we have also the long reads that are supported mostly by Oxford Nanopore and QuackBio. And as you guess, with this technology, we produce very long reads, very, very long. It can be several thousand of bases in one time. But the biggest drawback of this technology is that there is a lot of sequencing error inside of this sequence. And also the price is still expensive. It's more and more affordable, but it's still more expensive than short reads. So if you want to do the gene annotation with that, you will have to do first for both of technology 2 steps, which are the read quality check to check if there is no problem in the sequencing, in the quality of the basis, and also clean the reads to remove the adaptators, chimeric sequence, etc., etc. And once you've performed those two steps, the analysis will differ between these 2 technologies. So for the Illumina reads, for short read, you will align the reads along the genome. So they will align along the exons. And just after that, you have to do an assembly step. So you have to take all those reads and try to recompose, to build the puzzle and to regenerate the transcripts that have been that frame is originated. And once you have those transcripts, you can then define the gene annotation, the transcript annotation. So here you can find we have determined two isoforms for this gene. With long reads, it's very different because you align the reads, and because they are very long reads, you already have the gene intron structure. So there is less work to do, less computation to do, because you don't have to assemble anything. You just align, and then with the alignment, you just deduce the different transcript and gene structure. And as you can see, with the advantages of these long reads, it's more sensitive to isoform. It's easier to find the different isoforms. There is less error on this. So for the GeneSuite project, we developed 2 pipelines for dealing with the RNA-seq data and the ISO-seq data of the project. So my colleague from the INRAE of Toulouse developed the Takeda pipeline. And myself, I worked on the ISO-seq data, and I developed the NF-core ISO-seq pipeline. I won't go into detail, but it looks like this. As you can see, it is decomposed in 3 main steps. I don't know if you see my mouse, but on the left, you have the post-processing of the reads. So the quality check and the cleaning of the reads. Then the second step is the mapping. So you align the reads on the genome. And the first one is the generation of the transcript notation and gene notation from the alignment. So the advantages of this method is you have a very good detection of the isoform. You don't have any model to train. It tends to produce less false positive when you know the drawbacks of each technology. But the big disadvantage of these 2 methods, it will not produce an exhaustive gene notation. It will depend on when did you extract the imagine you just extract mRNA for one tissue at one time point. You will detect only the genes that have been expressed at this moment, compared to ab initio method, where they try to detect everything. So if you want to be exhaustive with this kind of annotation, you have to keep this in mind. You have to multiply the sample. The 2 other drawbacks of this method, it's the computation. As I said, the assembly step is very, very heavy, and also the alignment is pretty heavy to compute. So it can be long, do not forget that. And also the storage. For the ISO-seq, for example, in the GeneSuite project, we had 42 terabytes of raw data. Okay, sorry. yeah, so it's a lot of space. You won't store it this on your laptop. And so the last type of methods that exist, it's the pipeline methods, where it's pretty simple. You just take the ab initio, you take the homology methods, and you combine everything in one big pipeline. And the general methods for gene annotation, it's you first use the evidence to train a model. You generate an annotation, and then you use this annotation to train the, you train the model, and then you do the definite annotation. For presenting this, I just borrow the slide of Breaker 3, which is one of the most famous annotator for genome. And as you can see, this program takes as input some RNA-seq, so short read. It uses also some protein data sequence and the genome. And the first step is to assemble the RNA-seq into a transcript, then make a first prediction of genes with GenMark, then train GenMark again for producing different annotation. Those annotations are used to train Augustus and create a new annotation. And at the end, a program will try to take all these annotations that have been generated to create the definitive one. So as you can guess, this kind of method will produce the most exhaustive gene annotation, but it's very heavy to set up. You have a lot of installation to do. You need a lot of data. You need a lot of computing. You need a lot of storage. So yeah, it's good, but there is a lot of drawbacks. So in conclusion, which method do you choose for genome annotation? It depends on what are your resources and what are your objectives. If you want to just describe the gene of your genome, you have a new genome, you want to do a landscape of the gene inside. Maybe you can just do an ab initio method and you will have a good overview of it. But if you have a little bit more of budget, you can think about sequencing some RNA and improve your annotation. But if the objective of your project is to have the most complete annotation, you have to go to the pipeline. But don't not forget, it will ask you a lot of data and a lot of computing resource. So in the end, genome annotation, it's not a trivial task. You have to very well define your objective, to list all the public data that are available, define the data you will have to generate the sequence. Will it be short read? Will it be long reads? Check the available tool. There is new tools almost every year. And be careful with the informatic resources. And also, as I said at the beginning, there is genes, but they are not alone. There is a lot of other things inside of it. If you want to go a little bit further, or if you are very interested by the subject, you can try to read this publication. The first one is an introduction guides for genome annotation. The second one, it's a benchmark of the ab initio methods which is for the most recent of them. And the last one is very interesting because they are explaining the false annotations tend to spread from genome to genome during annotation. So yeah, if you want to go further, go grab those publications. So thanks to everybody, like always, thanks to all partners for all the work and the help they gave me. And if you want to talk more about this subject, you can contact me on any of those links. Choose the one you prefer. Thank you.
Thank you very much, Sébastien. It was very interesting. Do we have any questions from the audience? In the chat, maybe?
Yeah, it seems not.
No. So I'll wait another minute. Thank you. Can we ask to unmute? Is it okay? Yeah, good.
I have a question because I think earlier I saw that you also used string tie.
Oh, I didn't use it myself, but string tie is used in Tagada, if I remember well.
But were you able to, do you have any experience using it before?
No, that's the first transcriptomic project I work on. So I don't have any experience on RNA-seq using RNA-seq short reads for annotation. But after, maybe I can try to answer your question.
Yeah, because like right now I'm also analyzing an RNA-seq data, and I use string type for annotation, like sort of for the naming of the transcripts. But then when I did differential gene expression analysis using ball gown in DESEC2, my problem is the gene IDs are named like MSTRG, and not, I'm actually expecting for it to be assigned with a more you know, the gene ID. So I'm asking, like, if I would just like to ask if there is a way wherein I can assign gene names to these like generated names by string type.
Yeah, it's a very specific question. I would tell you there is always a solution in bioinformatics. Yeah. But right now, no, I can't give you an answer on such a very precise point. But after, maybe you can try to contact Sylvain Foissac from the GeneSwitch project that made the RNA-seq analysis. They integrate string type in their pipeline. So maybe they will have the answer for you. But I'm sorry. Moreover, with the problem with ISO-seq, you cannot do differential annotation, expression annotation with it, because it's a problem of long reads. It requires a lot of material for the sequencing. So for the ISO-seq, they pulled the four samples together. So I cannot do any differential expression on it. So they use RNA-seq. Mostly people that use ISO-seq use RNA-seq for doing the gene expression analysis.
Yeah, okay. Yeah, because like in my data set, some of them actually have gene names, but then most of them are having the MSTRG, which I think is the default nomenclature of string type. And then I'm just curious, like, if there is a way to, you know, assign the proper gene names for those, because I am also interested to know which specific genes are differentially expressed. So, yeah,
I see what you mean. After one trick maybe can work. It's to use. I use Tama in a pipeline. I use Tama to merge annotation together, and it keeps tracks of the origin of the annotations that have been merged. So maybe what you can do is to take Tama, merge your original annotation with your original ID, and merge it with your new annotation. And in the resulting annotation, we have the track of the IDs inside of it. So, but maybe it's a little bit complex. Maybe there is just one option in string type for resolving this. So,
yeah.
Sorry,
Okay.
I can do
more.
Yeah.
Thank you.
Yeah, but if you have any general question about bioinformatics or anything, feel free to contact me. I will be happy to discuss with you.
Okay. Yeah, can you also maybe send your email address?
Yeah, I think I'm going to put you on the presentation. It's sguizard@AD/AC UK.
Yes, we can provide the presentation after.
Yeah,
or the video will be online, I think.
Yeah, exactly.
Thank you.
Thank you for the question.
Thank you. We have a question from Mabel, and I think me too. I would like to know, how do you test for false positive? And I'll add to it, like, how do, in cases where there is like conflicting evidence with different sources, how do you deal with that?
So what did you see this question? It's in the chat? Because I have a question about the mono multi-exonic.
Yeah, just before that. Just before that. Yeah, Mabel.
Oh, test for false positive. Oh, it's pretty complex. I will talk about my annotation, because this is the one I know very well now. It has been pretty hard. I think it has been mostly the biggest things to clean for cleaning the annotation. The best way for removing the false annotation is to know well how your technology is working. For example, for ISO-seq, I found the biggest source of false positive is due to the primers that are being used for the library preparation. Because the primer used for ISO6, there is a poly stretch inside of it, which is supposed to attach at the poly tail of the mRNA. And so you sequence the complete mRNA like this. But if you have a poly stretch inside your mRNA, you will not do not sequence the complete sequence, but only from this poly stretch. And so by knowing that, I could search for annotations that had a poly stretch after the last exons and detect that those are false positive. And yeah, there's after you can also compare to the reference annotation, but it's complex because reference annotation, when I compare the ISO6 to the reference annotation, I see I have the double number of isoform. So how do I know my isoform are true? So you see, it's a pretty complex task. But yeah, start by knowing the technologies that are using and the drawbacks of this.
Thank you very much. Mabel, does it answer your question? Yes, thank you. So the next question is from Adela. I don't know if Sébastien or Ed want to go for it.
I like this question because I'm working on it actually. It has been very problematic also with ISO6 because I detected a huge number of monoexonic genes. And I think my intuition right now, I think is they are real. It's not false positive. They are not been annotated before. It's only theories. I'm working on it, so I have nothing. I'm not sure of anything right now. But those monoexonic genes, intronless genes, have a very short half-life, and I think the RNA-seq the coverage of this monoexonic is very low on RNA-6 method, but with ISO6 I authorize a very low number of reads. So I find them, and I think just RNA-6 just forgot them because they were considered as noise. So I'm working on it, but it's an open question right now. I can tell you which is the good ratio between multi-exonic and monoexonic because I think it's totally biased. I'm gonna try to have an answer on this. But as you can see, with the technology you have, I had to check if it was not false positive. I searched for months what could generate this, and my intuition now it's real result. So yeah, it's very complex. You have a lot of things to check.
Yeah. Maybe it's good to mention how long a project like this takes.
For me, since I started 2 years, but I developed also the pipeline. It has been very long for me because yeah, I had to develop the complete pipeline for ISO6. It's the ISO6. There is all the programs that are available, but they are nothing automatized. And depending on the strategy, people are not using the same strategy. For the pipeline, I use a strategy that do not follow the recommendation of PacBio. I stop before what they say, and so yeah, I had to develop the pipeline to test it and produce the result and restart the pipeline several times. It's all these things can be very long, so be careful. If you use something that already exists, it can be much more fast. If you just do RNA-seq and you use SAREC, NF-core, any NF-core pipeline that already exist, it can be done pretty quickly. But if you are on edgy things like I was, yeah, it can be pretty long.
Just another point of view, we started to build the project in 2017. So 8 years ago. Well,
exactly, yeah.
7 years ago. So it takes time to do it.
Exactly. I think that's
just to give an idea for the master students, the funding for this project is 6 million euros.
Yeah.
And the part dedicated to functional annotations and data sharing is more or less they have 2,500,000. No, what I said, yeah, 2 million, around 2 or 3 million.
Yeah, big funding for 8 years. Thank you both. Do we have any questions more for Sébastien, Hervé, or for Nathalie? So no, not anymore. I will share my screen.
Yeah,
now.
oh, sorry, I give you.
Thanks, Sébastien. Yes. So is it good? Yeah. Do we see? Yeah. So now we come at the end of this really interesting webinar. And I would actually like to invite all of you on the 4th of May for the genomic prediction. So it's the second episode of this webinar series regarding decoding genome in monogastric. And we will be very happy to see you again and to hear from you. And I hope we stay in touch. And if you want to know more about the Gene Switch project, you can contact the coordinator, Elisabetta. And regarding the EMABG program, Gabor is the coordinator, and you can also get in touch with him. And we put the QR code there, and you can follow us and subscribe to our mailing list, and you will receive more information about the series of webinar. And yeah, thank you, and do not hesitate to follow us and engage with our content. And yeah, thanks.